Hello, my name is Millican. Today I'm going to be talking about cellular respiration, which is the process of which all organisms get get energy, and obviously very important process. And there's two different types of energy production that we're going to talk about, or metabolism. Okay, first off, the first process is through anaerobic. Okay, anaerobic, as many of you probably know, is without oxygen. Okay, so it does not utilize oxygen, nor is oxygen a byproduct of this process. And there's a couple different processes that can be anaerobic. First off is glycolysis. Okay, now glycolysis can happen in all different types of organisms. Okay, so it can happen in prokaryotic, it can happen in eukaryotic, different archaea and bacteria, and even fungi can undergo glycolysis. Okay. And we'll talk about glycolysis in a bit. We'll go into depth because this is a very important process you need to know, even though you'll find out that it's pretty inefficient in creating energy. Okay. There's two different things that can come out of glycolysis. Okay, according to Deanie's video. Okay. Again, Holden Hall. They have the videos for cellular respiration 1 and 2 from Deanie's class if you want to go watch it. I'm creating this video in case you don't want to go watch it. And this way I can also learn it as well. Okay. By the way, let me add a point to it. Glycolysis is a very ancient process, okay? But it's been very evolved. It's alright ancient. I won't go into the history of it. If you want to find out about it, just go back to your lecture notes. Dean kind of talked about it. But one important thing to remember is that it's cytosolic. Okay, so it does not happen in membrane-bound organelles. Okay, it's membrane-bound in the fact that you have the double phospholipid bilayer, on the outside of the cell. But although not in bacteria or in prokaryotes. Okay, but it happens in the cytosol. Okay, and this is important because we're going to be talking about some processes that happen in the mitochondria, and that is not the same thing. Okay. So two different glycolysis procedures that can happen. Fermentation. Okay, many of you know fermentation from the creation of alcohol in plants. But one thing to remember, there's two different ones that you probably want to remember is lactic acid fermentation. Now, lactic acid is when we exercise and we're doing anaerobic exercise, obviously without oxygen, and you're getting sore and your muscles are cramping up, that's from lactic acid, okay? But there's also a different one, ethanol, okay? And we find that ethanol is in fungi, specifically with yeast, for bread production, for leavening your bread. Okay. The second one is cellular respiration. Okay. Actually, I think he was wrong. Glycolysis does not, cellular respiration does not, is not part of glycolysis, but it does. It's on the pathway, and we'll go over that. Okay. But cellular respiration can be anaerobic or aerobic, okay? Depending on the organism. Okay. And this also has the electron transport chains. And the thing to remember is that cellular respiration, especially with the electron transport chains, happens in membrane, okay? So happens in membrane, okay? And we'll go over that specifically with the mitochondria. Okay. So let's go over glycolysis, okay? Now glycolysis is a 10-step process. Fortunately, you don't need to know every step of the process, but we do need to know the gist. We do need to know what are the byproducts of glycolysis, okay? So we start out with glucose. Now, if you remember, glucose has 6 carbons, okay? And if you want to write the rest of it out, H12O6, okay? Now it's going to go, I'm going to draw 10 arrows, 3, 4, 5, 6, 7, 8, 9, 10, okay? Now the overall product of glycolysis are 2 pyruvate. Hopefully you can see it from here. Can you see it? Yes. And the thing about pyruvate is that now we have 2 of them. It's going to be C3. Okay? So we're not losing or gaining any carbons in this reaction, okay? But we still want to keep track of what we have. I think it really helps in learning this process, what goes where, byproducts and so forth, if we're going to really learn the process. So what happens? Well, there's kind of it's kind of a 2-step process, really. I know it's 10 steps, but it's really, there's one half and there's a second half. Okay, the first part is endergonic. Okay. To be honest, not exactly sure what that means, but he didn't mention the definition in the video, so I wouldn't be too worried. He also didn't mention lecture, as far as I know. You're going to have what we call dephosphorylation, okay? This is where you're losing a phosphate group on adenosine triphosphate, and we're going to turn that into adenosine diphosphate, okay? Okay? And that's going to happen twice. I'm not going to draw the whole thing, but you get the picture. In the second part, we have NAD positive, and that's actually going to gain a proton. If I can draw it right, DH. That's going to happen twice. And then, not to mention, you're going to have 4 ADP. And that's going to turn into 4 ATP. Okay? Now, obviously, you lost 2 ATP at the beginning. So 2 of these ATPs that you produce in the endogonic portion of glycolysis is just going to make up for the beginning, the inorganic portion of glycolysis. So really, you're just going to have a net gain, and I want you to remember this, you're going to have a net gain of 2 ATP. Okay, a net gain of 2 ATP from glycolysis. Also, you're going to have a net gain of 2 NADH. Okay? So we've just gained 2 ATP and we've gained 2 NADH. Now, I want to be very clear. Are we creating ATP? No, we are not gaining, we are not creating ATP. No, we are gaining ATP. We're going to go over the process of ATP production later on. Okay, that's important. Now, from the pyruvate, there's a couple things that can happen. Okay, but for one, okay, remember I mentioned ethanol fermentation, right? Well, it can go ethanol fermentation. We're going to go over what comes out of that. It's actually kind of cool. Now, we're still not talking about total or solely eukaryotic organisms. Ethanol fermentation happens generally in prokaryotic, but it's still important to understand, okay? So, you're going to have a waste product in fermentation of CO2, okay? And that's going to be 2 of them, okay? And what you get out of it is 2 aldehydes, which are C2 aldehyde. Okay? And you're actually going to not oxidize, but you're going to reduce. He kind of confused me a little bit in the video, but you're going to reduce it, okay, to 2-ethanol. Okay. Now, if any of you are in organic chemistry, you'll know that in hydrocarbons, 2 carbons use the prefix of ETH. So when we are writing ethanol, we write ETOH. Okay, so that way you don't get confused when I write ETOH. Now, ETOH, it's a waste product. Okay, so you're asking, why does it undergo this whole process of creating ethanol when all it is a waste? It's not totally a waste. Okay, because through this process, it's actually going to replenish. It's going to replenish the NADH. Okay, so it's going to recycle it. That way, it can go through glycolysis again. Okay, so that's actually pretty cool. This is all before we get to the mitochondria. Okay, so we're still outside the mitochondria. And we've already created 2 ATP, net 2 ATP, and we've also created 2 NADH, okay? And we'll go over what NADH does. He didn't really specifically say what NADH is, but it does have a specific purpose, and that purpose is mostly in electron transport chains, okay? So now let's get to getting into the mitochondria. I'm going to erase this real quick. Now, NADH has a hydrogen on it. So how is it able to get through the membrane when it has a net charge? We learned earlier that it can't just that ions can't just diffuse through the membrane, okay? So it's going to do something a little bit. I'm going to draw the mitochondria first. Okay, so here's the mitochondria. Okay. You got these little folds in here. Blah, 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 blah. Okay. I'm going to write out what's on here. So we got the outer membrane. We have the inner membrane. Inner for short, okay. There's an inner membrane space between the inter and outer membranes. I'm going to label it IMS, sorry. And then we have the cristae, which are the actual folds. Hopefully you can read that. Or you're looking at your notes, hopefully. Okay? And then inside we have the matrix. Okay? Now, why is this important? Well, there's going to be a few different functions that happen in the mitochondria. It's important to know the placement of it. And I'll mention when we go over the processes where those metabolistic processes occur. But what I was talking about earlier is that obviously every organelle in eukaryotic organisms in the cells have a phospholipid bilayer. Okay, so I'm going to draw that phospholipid bilayer, and I'm going to get a little bit of a space right here. Okay. Now remember we have pyruvate. Okay, so I'm going to, I guess, draw this pyruvate. Okay, it's C3. Okay. Now in the membrane, and Dini said in the video that they're not exactly sure how this occurs, but you have 2 different proteins in this membrane, and that pyruvate is actually able just to come straight through. He didn't say anything about it being gated or anything. He just said that the pyruvate is able to get through. And I'm going to write this 2 times just to remember that for every molecule of glucose, there are 2 molecules of pyruvate, okay, produced, okay? So basically, after glycolysis, everything is multiplied by 2, okay? So now we have the pyruvate in the IMS, okay? C3, okay? Now, when it's in the inner membrane space, it's going to lose a carbon, okay? It's going to turn into C2. Now, there's also a few things that will occur in that process, okay? Bouncing off of it, I'm going to get a different pen. Bouncing off of it, you have NAD+. It's going to turn into NADH, okay? We call this process pyruvate processing, okay? Now, how does it do that? Well, for one thing, you're going to have a waste product that comes off, CO2. That's where you're losing the carbon. Through pyruvate processing, there's a waste product of CO2 that comes off that pyruvate. And I'm not drawing out the full pyruvate. It would be C3H6O3, okay? So there's 2, it would be, you have CO2, so you have C2, you're losing a hydrogen right there and so forth. The way that it does this, it utilizes an enzyme, okay? Let me draw this. It's called CoA, or coenzyme A, okay? And like I said, this process is called pyruvate processing, but these 3 steps, the NAD plus turning into NADH, the waste product of carbon dioxide, and the utilization of CoA, this whole process utilizing those 3 elements is called pyruvate dehydrogenase. Okay. And this is what catalyzes the whole reaction of those 3 different steps, okay? And what you're going to have at the end of it, that C2 is called acetate. Okay? So now that we're in the membrane, in the inner membrane space, we lost a carbon and now we have an acetate, okay? Now why is this important? Well, it's going to join up with what we call an oxalate acetate. Okay? It's going to link up with that carbon 2 to create a citric acid, or a C6. Okay? So now we have 6 carbons. Okay? And this is where we start getting into the Krebs cycle. The Krebs cycle was named after a guy whose last name was Krebs. Yay! Okay. Now you're probably asking me, well, where did that oxalate acetate came from? It comes from the Krebs cycle. Okay, so I'm going to redraw this actually. Hopefully you get it. It looks like guys can see it, so I'm not going to get it up close. You can pause it if you want to see it. I'm going to redraw that process, or actually more expanding on it, and I'm going to talk about the Krebs cycle, okay, which happens in the mitochondria. Okay, remember glycolysis did not occur in the mitochondria, but this does. Okay, and this is in eukaryotic organisms. Okay, so now I have that C2, which remember is acetate. Okay, it's going to link up with a carbon 4 or a oxalate acetate. Okay. I'm going to draw a couple arrows with the citric acid. Okay. And when you're drawing the Krebs cycle, you can basically just draw a bunch of arrows like this. Okay. I'm sure you're laughing at those marker sounds, so you guys are so mature. Okay, so there's a couple different places where you're gonna have waste products, okay? So obviously, to get from C6 to C4, we're gonna have to lose 2 carbons, okay? And that's gonna occur like right around right here, CO2, okay? We're gonna also have another place right around here, CO2, okay? And around this area, I'm gonna get a different pen. Around this area, you have an NADH, or NAD plus, sorry. It's gonna turn into an NADH, okay? Gonna gain a hydrogen, okay? That's gonna happen again right after the CO2. So I'm gonna draw another arrow. So NAD plus turns into an NADH. Okay? Now around here, we got something that we haven't seen before. An FAD is an FADH, another high energy molecule. Okay? And last but not least, another somewhere around here. I'm not sure exactly where, but you got an NAD positive turning into an NADH. Okay? So now let's go over the entire output. Remember I said that we had 2 ATP net gain and also 2 NADHs that came from glycolysis. Well, now we've gained, actually I'm gonna draw this on the side here, the output, okay, so far, okay? Remember, this is Krebs cycle, okay, that happens in the mitochondria, okay? By the way, this is gonna happen twice because there's 2 pyruvate that turn into 2 acetate. Okay, so this process is going to happen twice for every molecule of glucose. Remember that. Okay, so in the beginning, during glycolysis, we had 2 ATP. We had 2 NADH. Okay. We also had 2 NADH that happened between glycolysis and the Krebs cycle during pyruvate processing. Okay, so I'm gonna put that right here. Okay, 2 NADH. Okay, that's in a separate category. Okay, and now I'm gonna multiply all this by 2. Okay, so we have 1, 2, 3 NADHs. But multiply by 6, so we have 6 NADH. We also have 2 FADH. Okay, and by the way, so just to make sure you guys know, that first part was glycolysis. This part was from pyruvate processing. Okay, and this last part is from glycolysis. Okay, also, if you remember correctly, okay, and I'm not sure I drew it in there. Let me make sure. Okay, did not draw this in here. One last bit to this. Sorry if you're like, he has no idea what he's talking about, and you're absolutely right. Okay, we have a bunch of adenosine diphosphates in the mitochondria, well, you're going to have an ADP plus a phosphate that's going to turn into an ATP molecule in Krebs cycle, okay? So you're also going to have, in the Krebs cycle, 2 ATP, okay? So this whole process is Krebs cycle, okay? Okay? By the way, all these processes are very inefficient, okay? And you're going to see why later on when we talk about 2 of the last steps, okay? Now, where does this happen? Well, by the way, just to let you guys know, the process of getting these ATP molecules is through a process called substrate level phosphorylation. Why is that familiar? Because we've been talking about substrates. Substrates are what go into the active site of the enzyme to change the conformation. Is it to change the conformation? No. It just fills in the active site in order to break down, like let's say that you have a glucose and a sucrose molecule stuck together, you put it into the substrate and out comes glucose and sucrose, okay? But we're gonna have something a little bit different, and I'll draw this out for you. Hopefully you guys can see it. I can't draw any lower than that. I might have to erase this. Okay, I'll erase it. I'll give it a second so you guys can pause it if you want. Okay, alright. Okay, I'm gonna leave this chart on the side there. Okay. These are all substrate level phosphorylation, okay? Remember, phosphorylation is the addition of a phosphate group. So ADP plus phosphate leads to ATP, okay? So we have our enzyme, okay? We got that kind of those 2 active sites, okay? Okay, this is how it creates ATP, not creates, how it leads, the phosphorylation creates ATP, okay? It's not the generation of that, but you have like this hexagonal deal linked to a pentagonal, okay? Linked to a kind of like these 2 circle deals, okay? These are the phosphates, and then you also have this chain right here, okay? There's 3 of them. So they're in the active site, and they're going to come together. You have the adenosine diphosphate and the this is called the phosphorylated substrate. The addition of these coming together is going to create ATP. Okay, so it's going to utilize an enzyme to come together and create ATP. Okay, that's how it goes from adenosine diphosphate to adenosine triphosphate. This is different than electron transport chain. Okay, so now that we have our output, this output is going to go into the electron transport chain. Okay, so let's talk about the electron transport chain. Okay, by the way, I'm basically combining 2 lectures, so if this feels really long, take a break. It is a really long process to go over. Okay. Where does electron transport chain occur? Well, it happens in the membrane of the cristae. Okay, so I'm going to draw the cristae. You have the fold, okay. And we have this green deal that represents an electron transport chain, Okay. Now remember I said, I didn't do the math, but I'm doing it right now. We have 10 NADH and 2 FADH. I think I've been doing stuff. It's FADH2. Okay. I think I was doing FADH. It's FADH2. Okay. Now, this FADH. Let me draw this better. Okay, it's going to donate a hydrogen to the electron transport chain. So now we have NAD, and this can go back into the Krebs cycle. Okay, it's recycled. Same thing's going to happen to the FADH2. Okay, it's going to donate its hydrogens. So we have FAD positive. Okay. And I'm going to draw a place around here. Hopefully you can see this. It's recycled. Okay, no longer needed. All it wants is the hydrogens from there. Okay, by the way, just so we keep organized, this right here in the inside is the inner membrane space. Okay, hopefully that's right. Yeah, outside here is the matrix. Okay. And obviously this right here is the cristae. Okay. That purple one right there. Okay. So it's going to go in. And the carriers, maybe I should write this down. I'm going to write this. The carriers, these being hydrogen carriers, the carriers will transfer not only hydrogens, but also electrons. I'm going to put E negative and S. Electrons, by the way, when I say the electrons, I'm saying the electrons from the hydrogens, okay? Because obviously, like, there's not, like, electrons on there, they will transfer the electrons from the hydrogens to chemicals, to chems, I'm going to put, that make up the ETC, the electron transport chain. Okay. So what exactly is in this electron transport chain? Well, there's 4 different complexes. Obviously, I mean, like, there's thousands upon thousands of electron transport chains in this cristae. But in every individual electron transport chain, okay, there's 4 different, I'm going to draw like globs, okay? Okay. And obviously, in between here, is a phospholipid bilayer. Okay, this is going to be kind of important. Everything is important. By the way, if you have any questions, don't be afraid to hit me up. And if you have anything like, I don't think that's right, definitely hit me up, because I want to know if I'm wrong about any of this as you watch me wantingly draw phospholipid bilayer. Okay. And let's label these, okay? We got 1, 2, 3, 4, okay? Now inside every single complex, there are, I'm gonna write this here, 9 proteins and what I'm going to call 1 Q. Okay, a Q is also known as a ubiquinone, and they're pretty prevalent in the mitochondria, but I'm going to call it 1 Q. We're going to talk about the purpose of that ubiquinone in a bit. Okay. So we have these H positive ions with the electrons in there. Okay, I'm going to draw like a bunch of H positive. Okay? Now obviously they need to get, they're going to want to get up in here, but they can't go past the phospholipid bilayer because they're ions. So they're going to get helped out a little bit, okay? They will, they're going to get past up here through the, with the help of the eukarytone, okay? So they get up in here, and they're gonna, and these complexes are gonna pump hydrogen protons out, okay? And this is gonna happen every single place. Now, what happens to the electrons? Well, the electrons are gonna get transferred from one complex to the next, okay? As it does that, in order to get from one complex to the next, it's going to lose energy. So it's going to need help a little bit on the way. That's why you have the ubiquinone, okay? The ubiquinone is going to help it get, and let me make sure I'm telling you this right. Ubiquinone, okay, let me clarify, okay? The high energy electrons are going to help pump hydrogen ions across the membrane. The electrons being the hydrogen positive ions. Okay. The Q accepts protons and electrons from complex 1 and 2, and it easily shuttles it through the hydrophobic interior of the inner membrane. That's how the electrons are getting through here, okay, to the other complex. Why is it that the eukarytone can do this? It's because the eukarytone is very hydrophobic, okay? And the Q dumps the H positive into complex 3, which then goes into the interior membrane system, okay? This being the matrix, okay? That's going to be very important later on, okay? A part of a really cool process, okay? It's a little bit confusing, but you just got to know, with the help of electrons, it's going to help pump these hydrogen ions into here. And as the electrons also move from complex to complex, they get weaker until there are pretty weak electrons in this final one, okay? Before I jump ahead of myself, these electrons are now going to become waste, okay? And what's going to happen? Well, we have oxygen, O2, that's going to accept the low energy electrons to a very electronegative chemical, which is oxygen. By the way, in mitochondria, it's only oxygen that's going to accept these hydrogens, these low-level hydrogens. Okay, in bacteria and prokaryotes, it could be a nitrate or other chemicals that are less electronegative. But for mitochondria, it's always oxygen, and this is going to make water. Okay, so now we got the hydrogen coming in, so now we have H2O, and this is a waste product. Okay, after it's lost a lot of its energy, because it's losing energy as it uses energy to move from complex to complex. Okay, so so like I said, the oxygen is accepting the hydrogen ions, okay, and the electrons from complex 3 go into the electron 4 into the proteins, and the electrons pump more hydrogen into the inner membrane space, and the electrons are finally accepted by the oxygen, okay? And this creates an electrochemical gradient through concentration and also by charge, and even it's a pH gradient, okay? But none of this, again, I'm going to stress, just like I did with glycolysis and Krebs cycle, none of this creates ATP, okay? None of it. You know, obviously there's some process of, you know, the phosphorylation, dephosphorylation, but this does not create ATP, okay? First off, before I go into the last step, yay, the last step, because we have all these hydrogen ions up here, the intermembrane space is more acidic than the matrix, okay? And that's going to be really important. Obviously, you want to create, you know, through the process of osmosis and trying to keep a balance, this is going to create an interesting dynamic for the last step, which is ATP synthesis. And it's going to do that through a process called, or through, not a process, it's going to do it through this tool called ATP synthase. So let me draw this out. I promise you we're getting close to the end. So hold on. Okay. And he didn't draw it in class. He just showed the textbook. I'm actually going to draw it. I think it's helpful. Okay. So we have this green deal right here, okay, kind of this globule. Okay. Actually, sorry. Okay. And you have this like tubule coming out, and it's going to be at the bottom. This is called the Slater. I know it doesn't make sense until I draw it, but there's like a tube, okay? Okay. And then you have this cylinder-like deal, so. Okay. Okay, let me draw it a little bit less here. Sorry about this, guys, but I feel like this is really gonna, at least it helped me visualize it. Okay, there's this like darker area. Okay, this is where that cylinder ends. Okay, there's also, I'll draw it in black so I can differentiate. Okay. It's still part of it, it's just like an overlap, so I'm trying to help visualize it. Okay. And then obviously like these, not obviously, but like, okay. So, let me go over the different parts. So, obviously, like I said, you got the Slater, okay? This right here, I'm not gonna draw the other side of the phospholipid so that you can see the labels. Here, by the way, you probably couldn't see it before. Here it is. You got the FO unit, and you have the F1 unit. Okay, and then in between here, you have the rotor shaft. This is actually like, I'm not like being sarcastic, this is a really cool process. It really, not to get religious or anything on you guys, for those who aren't religious, it really makes you appreciate God's intricacies, okay? Because basically he created an energy turbine in the mitochondria, okay? So we have all of these H positive ions, hydrogen positive ions, okay? And it wants to get back, to balance it out, it's going to want to get back into the matrix. In order to do that, it's going to go through this little tunnel here, okay? And through that tunnel, it's gonna make this F0 unit spin. And by doing that, it's gonna make the rotor shaft spin. Now, why is that important? Well, because at the very bottom here, I'll draw it out. You got ADP. Hopefully you can see that. Yeah, you can. Okay. Plus PI. Okay, the phosphate. Put in ATP. Okay. When the rotor shaft changes, it's going to the protons in the ATP synthase is going to undergo conformational change in its shape that drive ATP synthesis from the ADP. Okay, it's not phosphorylation, it's ATP synthesis. Okay. Now, like I said, the H positive ions go down through their gradient from high concentration in the IMS to through a pore that's right here. Okay, there's a pore, and because it can't go through the phospholipid bilayer, and through the ATP synthase, and that's what causes the spinning, both of the FO unit and the rotor shaft, and then it's going to spit out ATP. So that's how ATP is created. Okay, and obviously he talked about how, and I won't draw it out. You just look in your notes if you want. How actually I will. I won't be lazy. The way that the ATP gets out. Okay, we have this is the where the membrane of the mitochondria is. You got, it's an antiporter where you have ATP coming out, and then when it's used up, ADP coming back in so that it can be synthesized again into ATP. Pretty darn cool process. Okay. So, I'll let you look at it, and if you want to pause it and draw it, you can. And then I'll go over the entire process in a general view one last time. So, pretty cool process. So let's go over the whole thing one last time. Okay, not like all 47 or 48 minutes, however long this has been. It's by the way, this whole deal of the recycling and the Krebs cycle and stuff like that, NAD turning into NADH and vice versa. It's all called feedback inhibition. Very important process. But let's go over the entire process one last time. So you start out with glucose, okay, through the process of glycolysis, which is the breakdown of sugars, leads to 2-pyruvate. It's going to go into the mitochondria through those proteins and do pyruvate processing. By the way, remember 2 ATP and 2 NADH come out of glycolysis. So we have pyruvate processing, and under that process we have, by the way, I'm going to draw 2 ATP and 2 NADH. Okay, through pyruvate processing, you're also going to have 2 NADH and no oxygen
or not ATP, but to CO2. And that CO2 is important because that oxygen is going to be the hydrogen acceptor, that low energy electron acceptor, because it's electronegated. It wants to fill its octet. And it's going to go, and this is in, it's going to go into the matrix. Okay, I don't think I said that before, but it's going to go into the matrix. Okay. And it's going to turn into, remember the acetate, but 2-acetyl. And it's going to use that coenzyme A in order to catalyze that reaction with the NADH, the carbon dioxide, and the 2-acetyl, which is C2. Okay, it's going to combine with the oxalate acetate from the Krebs cycle in order to create citric acid, right? So I'm going to draw this right here. Okay, the citric acid cycle, otherwise known as the Krebs cycle. Okay, that's another name for it. That might help you remember that it's C6 that it combines with that C4, and out of it's going to come 4 carbon dioxide. Okay. Also 2 ATP. Okay, and of course 6 NADH and 2 FADH2. Okay, you see that? Yay! Okay, that's not awkward. Okay, and then finally, or not finally, I guess, it's going to go over here. You got those 4 globules. Okay, the 4 protein complexes with 9 proteins and 1 Q or ubiquinone. Okay, and you're gonna have because I'm being lazy, I'm just gonna draw it, but you get the idea. It's a bilayer. You got the hydrogen on the outside. By the way, these are going to lose their hydrogens, but in the whole process, let me circle these. Okay, so we have the 2 NADH, 2 NADH, and 6 NADH. Okay, so coming over here, 10 NADH. You're also going to have 2 FADH, and then last but not least, you're going to have 4 ATP. So like we got 4 ATP that come from the glycolysis and Krebs cycle. I didn't mention this before, but I'm going to mention it now. Really important. After the electron transport chain, which doesn't create ATP, but through ATP synthase, what you're going to have, and I'll draw that last kind of like the spindle deal with, so this is the, I know it looks bad, but ATP synthase, it's going to come out. It's actually going to produce 25 ATP. So the max yield, okay, so I'm going to write this down right here. Max yield of cellular respiration, just metabolism. I'm going to write metabolism. Is 29 ATP. Okay, 29 ATP. And according to Dini, that's a 64% of the energy in glucose potentially, and that's a pretty efficient process. 25 ATP is a crap ton. The 2 ATP in each of those processes, that only counts each of those account for 3.4% of the potential energy out of this. In this process, we said that glycolysis and Krebs cycle or citric acid cycle are substrate phosphorylation. The ATP synthase occurs through oxidative phosphorylation. And I guess I should spell it out because I've been saying it a lot and I don't think I've spelled it out yet. So, oxidative phosphorylation, okay? And which makes sense because all of these hydrogens, you're losing the hydrogens on the NADH and the FADH, and that's how the ATP is created through the hydrogen going down its gradient in the ATP synthase to get the turbines moving, the shaft spinning clockwise or counterclockwise in order to change the conformation of the protons in, or is it proteins? Can you make sure that's right? Protons in the to change shape and drive ATP synthesis of the ADP, 25 ADP plus 25 phosphates that lead to 25 ATP. And that's how we create our energy. So a max yield of 29 ATP. And like I said earlier, that ATP goes out of the membrane through an antipore with the ADP coming back in order to restart this process. And obviously, that FAD and the NAD, those two products, are recycled back into the citric acid cycle. And if I'm not mistaken, part of glycolysis as well, because it's got to turn into, well, never mind, because it's got to, in the endogonic phase, it goes from NAD to NADH. Okay? And make sure I'm telling you right on that. Sorry, I'm sure this is really boring, but I want to make sure I'm telling you guys right. Yeah, so it goes from NAD to NADH. So it's going to get recycled for both glycolysis and the citric acid cycle. So that's it. That's cellular respiration. That's 2 lectures right there. And guess what? I covered it in less time than he did. So let's go. But if you have any questions, totally hit me up, and hopefully I'll be able to help you. One last thing. In the Krebs cycle, right here, I'm just going to draw a circle. 2 things, I forgot. 1, those hydrogens need to be oxidized, so they will, the hydrogens from the NADH and the FADH, so they will be donated to some enzyme and some coenzymes. Okay, you mentioned that in the video. Second, when I drew that cristae, let me draw it one last time. Okay, we had the green electron transport, and it goes throughout this deal. Okay, there's thousands of them. Okay. You have these little itty bitty dots, okay? Right? And that is where the ATP synthase is, okay, in between these electron transport chains. They are not part of the electron transport chains, but they are in the membrane of the cristae, okay, with this being the IMS and this being the matrix, okay. So, again, if you have any questions, hit me up, and hopefully this helped you out. I'm going to leave it for 1 second. That way, if you want to pause it and write some of this stuff down, or if you don't want to, that's totally fine. I understand. But hopefully this helped, and maybe I'll do another video.